Welcome to the OLSA Medical AI Podcast, where research meets reality and intelligence transforms healthcare.
Glad to be here for another deep dive into the sources you've all shared with us.
We have a really fascinating stack of research papers and clinical notes today. We are unpacking a completely new frontier in medical research.
Yeah, it's a dense one, but incredibly important.
It brings up this really bold question for the future of healthcare. What happens when artificial intelligence stops just crunching our numbers and actually becomes an autonomous co-scientist?
Right, an AI that actively designs experiments and makes biological discoveries all on its own.
Exactly. And just as a quick reminder, we are here to impartially report on the content provided in these sources. We aren't endorsing specific viewpoints, just breaking down the science for you.
And today's science revolves around a groundbreaking AI framework called Eubiota.
Eubiota. And it's designed to unravel one of the most chaotic, complex systems in the human body, which is the gut microbiome.
Everyone listening to this has a gut microbiome.
Right. You have billions of bacteria in your gut right now.
Trillions, actually. And it's a highly unique ecosystem. It dictates your immune system, your mood, your metabolism.
Even your risk for various diseases.
Yeah. And if you've ever felt, overwhelmed by how much we still don't know about gut health, just imagine how overwhelmed the actual scientists feel.
So true. And that is exactly what we are getting into today, how AI is finally stepping in to solve this massive puzzle.
Because it is a phenomenal challenge. The sheer volume of biological data we are generating globally it's completely outpaced our human ability to process it.
We know the microbiome regulates host physiology. It touches cancer progression, inflammatory disorders, the gut-brain axis.
But discovering exactly how it works, what researchers in your sources call mechanistic discovery, has essentially been stuck in the past.
It's highly manual, right?
Very manual. The workflows are fragmented across entirely different scientific disciplines. It is painfully slow.
The friction in traditional science seems to be a huge part of the problem here.
Oh, absolutely. Human research is incredibly siloed.
Like, a geneticist studying bacterial DNA might publish a brilliant paper, but then a metabolic chemist looking at gut inflammation might never even read it.
Right. So to make a real breakthrough, a human researcher essentially has to read thousands of papers across all these different disciplines.
Manually integrate all those hypotheses.
Design an experiment and just kind of hope they connected the dots correctly.
Which is why we've been trapped in this era of simple correlations.
Exactly. A traditional study might say, this specific strain of bacteria is present when a patient has a certain disease.
That's a correlation.
Yeah, it's a starting point, but it's not a cure. What doctors and hospitals desperately need is a mechanistic framework.
They need to know the specific causal chain.
They need a roadmap that says this exact bacterial gene produces this specific molecule, which then triggers this exact inflammatory reaction in your gut tissue.
But finding that precise chain among billions of biological variables, it's like finding a needle in a haystack.
Except the haystack is constantly changing shape and you're blindfolded.
Okay, let's unpack this. Enter Eubiota.
Yeah, Eubiota.
The sources describe it as an open-source, modular agentic framework.
Which sounds like a mouthful, but it's actually a very elegant concept.
The easiest way to conceptualize this is to think of a human research team. Because until very recently, a lot of scientific AI was built as one giant monolithic model.
Right, a single know-it-all brain.
And the inherent flaw with a monolithic AI is that when it doesn't know the answer to a really complex question, it tends to just make something up.
It hallucinates.
Exactly. But Eubiota doesn't work like that. It's a modular team of specialized AI agents.
All working together on a single project, actively checking and balancing each other's work.
That modularity completely changes the paradigm for medical research.
It really does. Because in a monolithic model, the planning, the data searching, the writing, it's all mashed into one opaque process.
You can't see how it got the answer.
Exactly. But Eubiota delegates tasks.
Yeah.
It mirrors how a real-world principal investigator runs a biological lab.
You have one person planning the overarching experiment.
A technician running the specific assays.
A quality control expert checking the data.
And someone synthesizing the final report. Eubiota digitizes that exact, rigorous human workflow.
The architecture here is just brilliant, so we need to look at how these four specific agents interact. First, you have the planner.
The boss of the operation.
Right. You hand Eubiota a highly complex scientific query, and the planner decomposes that massive question into a multi-step strategy.
It defines the specific goals and selects the exact tools needed to achieve them.
And once the strategy is set, the planner hands the instructions off to the second agent in the framework, which is the executor.
The executor is your highly skilled, tireless lab technician.
And Eubiota equips this agent with live access to 18 different specialized tools.
To really understand the power of this, look at the databases it can pull from. It has access to PubMed,
Which is basically the world's biomedical literature.
Right. It also uses 𝗏𝗃 biological databases.
For those unfamiliar, 𝗏𝗃 is essentially this massive mapping system for understanding high-level cellular functions.
From genomic information, yeah. And the executor also taps into MDIP.
The microbe disease interaction phenotype database.
Right, a tool that maps which specific bugs correlate to which specific physical symptoms.
It can even search internal lab data to look at raw wet lab results.
And it utilizes a Python coder to run its own quantitative analyses right on the fly.
So the executor takes the planner's intent and translates it into strict, validated commands for all these tools.
Enforcing the syntax so the AI doesn't just crash the system when running these massive queries.
But once the executor gathers all that disparate data, it doesn't just blindly pass it along.
No, it hands the raw findings over to the third agent, the verifier.
The quality control department.
Exactly. The verifier rigorously audits the evidence, it checks for logical consistency, and actively hunts for contradictions in the literature.
It basically asks, did we actually answer the boss's question with verifiable proof?
And if the evidence is weak or contradictory, the verifier rejects it. It tells the planner to try a different search parameter.
It only stops the process when a critical mass of solid verifiable proof is gathered.
Which brings us to the fourth agent, the generator.
The communicator.
Right. It takes all those verified notes and writes the final evidence-grounded answer.
Complete with perfectly formatted citations.
And what keeps this entire multi-agent team functioning smoothly is something the researchers call a shared structured memory.
Which is such a cool concept. Picture a digital whiteboard sitting in the middle of a laboratory.
Every single plan formulated, every database search executed, every piece of evidence gathered, it's all logged on this whiteboard.
Every verification check, too.
Yeah. This is a critical component because it prevents the AI from getting stuck in an endless loop or just repeating the same mistakes.
If the executor runs a search on a specific protein and it comes up empty, that failure is logged in the shared memory.
So the planner immediately knows to try a different approach rather than banging its head against the wall.
Here's where it gets really interesting. How the researchers actually trained this AI team.
Because you can't just plug a group of agents together and expect them to instantly understand the nuanced biology of the human gut.
No. And instead of forcing the AI to perfectly mimic a human's step-by-step logic, which is how most traditional models are trained, they used a reinforcement learning strategy.
It's called GRPO-MAS.
Group Relative Policy Optimization for Multi-Agent Systems.
Right. And the best way to understand GRPO-MAS is to imagine teaching a kid to solve a complex maze.
The traditional way to train an AI is through supervised, rigid micromanagement.
You'd stand over their shoulder and say, take a left here, take a right there, stop, turn around.
But with GRPO-MAS, you remove that micromanagement.
You place the AI at the start of the maze and simply say, I will reward you when you reach the exit.
You allow the system to explore and to fail.
And the implications of this training method are profound. By letting the AI navigate the complexities of biological data on its own, the Eubiota planner learns emergently.
It figures out its own logic pathways.
It learns that if a specific keyword search in the 𝗏𝗃 database fails, it needs to self-correct and try a synonym.
Most importantly, it learns to independently prioritize hard mechanistic data over superficial correlative data.
Because the mechanistic data is what ultimately leads to the correct scientific conclusion and the reward.
The results of that specific training approach are just staggering. Looking at the benchmark data provided in your sources, they tested Eubiota on highly complex biomedical reasoning tasks.
And Eubiota achieved an 87.7% average accuracy.
To put that in perspective, they ran the exact same test on frontier AI models. 𝗏𝗏𝗃𝗏𝗃 scored 67.2%.
And the proprietary GPT 5.1 scored 77.3%.
So Eubiota outperformed them by a really wide margin.
But they didn't stop at automated benchmarks. They conducted a blind test utilizing human domain experts in microbiology.
When these actual scientists compared Eubiota's answers side by side against GPT-5.1, Eubiota won the human preference 70.6% of the time.
And that overwhelming human preference really comes down to the necessity of trust in science.
A general-purpose AI might generate a beautifully written paragraph about gut bacteria.
But to a working scientist, if that paragraph lacks verifiable citations and a clear, logical trace of exactly how the AI arrived at its conclusion, the answer is functionally useless.
It has to show its work.
Exactly. And Eubiota provides an exact reasoning trace. It shows the human researcher every single paper it read, every database it queried, and the exact logical steps it took to reach its hypothesis.
We have established this incredible architecture, but the most important part of your sources details what Eubiota actually accomplished in the real world.
The proof is in the pudding.
The paper outlines 4 distinct end-to-end case studies where Eubiota drove actual biological discovery. Let's look at case study 1, genetic discovery.
The human researchers wanted to understand how certain gut bacteria managed to survive in an inflamed gut.
Because when your gut tissue becomes inflamed, your immune system releases nitric oxide,
which creates something called nitrosative stress. It essentially turns your gut into a highly toxic environment.
The researchers had this massive data set containing almost 2,000 different bacterial genes.
And they tasked Eubiota with determining which specific genes allow bacteria to survive this toxic stress.
This is a perfect example of a problem where human cognition becomes the bottleneck.
Analyzing 2,000 genes across multiple databases manually would take months.
But Eubiota took that data set, queried the genomic databases, synthesized the relevant literature, and correctly identified the 𝗚𝗲-𝗪 DNA repair axis as the critical survival mechanism.
It logically deduced that in a highly oxidative and nitrosative environment, the primary threat to the bacteria is severe DNA damage.
Therefore, the bacteria possessing robust DNA repair mechanisms are the ones that persist.
But this wasn't just a theoretical prediction on a computer screen.
No, the human researchers took Eubiota's hypothesis into the physical lab. They created mutant strains of a bacteria called 𝗏𝗏𝗪𝗏,
specifically engineering them to lack those exact genes.
And when they exposed these mutant strains to nitric oxide on the lab bench, the bacteria died.
Eubiota autonomously predicted a novel biological mechanism, and the physical reality validated it perfectly.
It's amazing. Case study 2 takes us a step further, moving from genetic discovery to actual therapy design.
The researchers tasked the AI with designing a synthetic microbial community, often called a 𝗏𝗏,
to treat colitis, which is severe inflammation of the colon, in a mouse model.
Currently, the medical field relies heavily on fecal microbiota transplants for severe gut issues.
But an FMT is highly unpredictable because you are essentially transplanting an entire unmapped ecosystem from one person to another.
A defined synthetic community built from scratch is much safer.
So to build it, Eubiota had to logically debate between creating a bacterial community that produced acetate versus a community that produced lactate.
This requires the AI to navigate a very real, ongoing debate within the scientific community regarding the specific roles of these short-chain fatty acids in gut inflammation.
And Eubiota evaluated the vast literature and made a highly strategic decision to deprioritize the acetate-producing community.
The reasoning was brilliant. The available acetate-producing strains included a specific microbe called 𝗏𝗏𝗪𝗏𝗏𝗏𝗏𝗏.
And Eubiota's shared memory recognized from the literature that 𝗏𝗏𝗪 is strongly associated with pro-inflammatory dysbiosis.
The AI realized that even if the metabolic output of acetate was beneficial, introducing a known pro-inflammatory bug into an already inflamed colon was a dangerous therapeutic choice.
Therefore, it successfully proposed a 4-strain consortium focused heavily on beneficial anti-inflammatory bacteria,
including 𝗏𝗏𝗪𝗏𝗪𝗏𝗏 and 𝗏𝗏𝗪𝗏𝗪𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏
bacterial cocktail in the laboratory and administered it to mice suffering from severe colitis.
And the mice, given the AI's custom cocktail, showed significantly reduced colon inflammation.
They experienced far less weight loss compared to the control group, too.
The AI autonomously designed a living biological drug that worked in a living organism.
What's fascinating here is how Eubiota applies logic across entirely different biological scales. In case study 3, we see it move into pharmacology.
The researchers asked the AI to design a commensal-sparing antibiotic cocktail.
Broad-spectrum antibiotics are notorious for decimating gut health.
Right, they act like a bomb, killing the beneficial commensal bacteria right alongside the dangerous pathogens.
So Eubiota was given the task of finding a precise combination of drugs that would eradicate dangerous pathogens, specifically Pseudomonas aeruginosa.
while leaving the healthy gut microbiome intact.
Eubiota mined the chemical interaction databases and designed a highly specific three-drug cocktail consisting of piperacillin, tazobactam, and amikacin.
And when the lab tested that specific three-drug combination, it worked exactly as designed.
It successfully separated the targets, neutralizing the pathogens while preserving the beneficial commensal bacteria.
Moving into case study 4, we see Eubiota tackle molecular discovery.
Here, the AI was let loose on human dietary intervention data, specifically analyzing untargeted metabolomics data from human subjects on a vegan diet compared to an omnivorous diet.
Untargeted metabolomics is essentially taking a massive chemical snapshot of every single molecule the body is producing at a given moment, without looking for anything specific in advance.
It generates an ocean of noisy, complex data that is incredibly difficult for humans to parse.
But Eubiota analyzed this massive snapshot and discovered that certain specific metabolites associated with the vegan diet actively suppress inflammatory signaling in the human body.
Most notably one called 3-methoxycatechol.
Right, it specifically identified the suppression of the NF-kappa B pathway.
To put that in accessible terms for you, the NF-kappa B pathway is essentially the cellular fire alarm in the human body.
When it gets tripped, it triggers a cascade of inflammation.
And Eubiota found the exact molecule in the vegan diet that helps silence that specific fire alarm.
When you step back and look at the sheer scale of what this system accomplished across these 4 distinct case studies, it is breathtaking.
During these runs, Eubiota analyzed over 10,000 individual scientific papers.
10,000. The research team calculated that the AI is 38 times more efficient than a human domain expert performing manual curation.
If a human scientist spent just 3 minutes rapidly skimming each of those 10,000 papers, it would require over 500 hours of continuous, unbroken reading.
And Eubiota processed the data, synthesized the findings, and provided perfectly formatted, highly accurate citations in just over an hour.
This level of capability sounds incredibly futuristic, but we need to ground this in reality and discuss the limitations outlined in your sources. Eubiota has not completely replaced the human scientist.
No, the framework is fundamentally reliant on a human-in-the-loop process.
Let's look back at the antibiotic cocktail from case study 3 for an example.
When Eubiota initially proposed the drug concentrations for piperacillin and tazobactam, it suggested a level of 100 micromolar.
But when the human lab technician actually ran the physical wet lab assay, they observed that at 100 micromolar, the solution was slightly toxic to the beneficial 𝗥𝗏𝗄𝗏 bacteria.
Which is precisely where the collaborative nature of this technology shines. The AI did not know the concentration was toxic until the human lab worker took that physical reality check and fed the wet lab data back into Eubiota's shared memory.
The human essentially said, hey, this concentration is killing the commensals.
And the AI's planner agent received that feedback, recalibrated its internal logic, and reduced the dosage proposal down to 50 micromolar.
That revised dosage ended up being the perfect therapeutic sweet spot.
So the AI is brilliant at synthesis, but it absolutely requires the physical grounding provided by human researchers.
That feedback loop highlights its most fundamental limitation, right? Eubiota is entirely bound by existing literature and documented databases.
It is a master synthesizer and an unparalleled logical inference engine.
But if a biological mechanism has never been documented anywhere, or if it cannot be logically inferred from existing published data, Eubiota cannot pull it out of thin air.
Exactly. It cannot physically operate a pipette to generate completely novel, raw data on its own. It operates within the boundaries of recorded human knowledge.
However, when we talk about the practical deployment of this technology, Eubiota solves a massive problem for clinical adoption.
One of the biggest hurdles for integrating AI into medicine is privacy.
Hospitals cannot simply upload highly sensitive, personalized patient microbiome data to a public cloud server to be analyzed by a commercial AI model.
It's a regulatory nightmare.
So what does this all mean for the actual hospitals and research labs?
Well, the creators built Eubiota so that it can operate locally, entirely behind institutional firewalls.
It can run directly on a hospital's secure internal servers.
This protects patient data sovereignty, which is a massive practical win for real-world feasibility.
A hospital can utilize the power of a multi-agent AI framework without ever risking a patient's protected health information.
If we connect this to the bigger picture, the 5-year vision for this technology is profoundly disruptive. but in a highly collaborative way.
Eubiota is not designed to replace clinicians or researchers.
It is fundamentally engineered as a co-pilot. The true value of a human scientist lies in high-level strategic thinking, intuition, and the ability to physically validate concepts in the real world.
By freeing humans from the grueling, nearly impossible task of reading tens of thousands of fragmented papers, Eubiota allows scientists to spend their time actually doing science.
Imagine what this means for you as an individual navigating your own health.
Right now, if you experience a gut issue, a doctor might recommend a generic, off-the-shelf probiotic.
But in the near future, systems like Eubiota could look at your unique gut genetics, your specific dietary habits, and your transcriptomics.
Which is the study of how all your genes are actively expressing themselves right now.
And the AI could autonomously design a hyper-personalized probiotic consortium or a highly specific antibiotic cocktail tailored perfectly to your unique biology.
We are witnessing a fundamental paradigm shift in medicine here.
For decades, the medical field has been forced to treat the human gut microbiome like a black box.
We put interventions in, we observe what happens, but we have never fully understood the intricate wiring inside the box.
Eubiota represents the exact moment we transition from merely observing that black box to actively reverse engineering it.
We are finally beginning to understand and manipulate the microbiome as a precise, predictable biological machine.
It provides a phenomenal blueprint for the future. It proves that when we align artificial intelligence with the rigorous step-by-step logic and verification processes of the scientific method, the results are deeply grounded.
They are highly auditable and immediately actionable in the real world.
The research on Eubiota clearly demonstrates that the future of medical discovery is not just about building bigger, more data-hungry, monolithic AI models.
The future is modular, multi-agent AI. By breaking the scientific method down into discrete agents that plan, execute, verify, and generate, Eubiota successfully turns an overwhelming, chaotic ocean of fragmented biological data into executable, life-saving laboratory protocols.
It's a huge leap forward.
We really encourage you to stay curious about this framework. If you are intrigued by the hidden mechanics of your gut or how artificial intelligence is evolving beyond simple chatbots, we highly recommend exploring the Eubiota research provided in your sources.
It's definitely worth a read.
Keep an eye out for our next deep dive as we continue to extract the most important insights from the materials you share with us. This was the OLSA Medical AI Podcast, decoding intelligence in medicine, one breakthrough at a time.
But before we go, we want to leave you with one final provocative thought to mull over.
We have just explored how an AI system can now autonomously formulate a complex biological hypothesis, read 10,000 papers to verify it, and design the exact physical protocol needed to test it.
This means the primary bottleneck in scientific discovery is no longer the processing capacity of the human mind.
The bottleneck is now simply the physical speed of the laboratory equipment in the human hands pipetting the liquids.
So moving forward, will the biological laboratory of the future even need human scientists at the bench?
Or will the lab eventually become a room full of automated robots silently executing the daily recipes drummed up by our AI co-scientists? Something to think about.